Welcome everyone to the 8th webinar of the Science of Aging Series 2021, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. For this series, we've lined up a number of webinars, all focused on the science of aging being conducted by leading researchers around the world in their relevant fields. This is Sarah McFarlane from Inside Scientific, and I'm very pleased to be your host for today's event. Today's webinar is titled Aging and Skeletal Muscle Plasticity and will feature Dr. Sue Bodine, professor in the Department of Internal Medicine at the University of Iowa, Carver College of Medicine. In her presentation, Sue will discuss her research focused on the decline in muscle mass with aging and the role that innervation plays in muscle atrophy. Before we get started, we'd just like to acknowledge our partners at the APS and the Alliance for Aging Research, and also to thank the sponsors for this webinar series for making it all possible, and a specific mention to Aurora Scientific, who has sponsored this particular lecture. So without any further delay, I'm very pleased to welcome Sue Bodine. Sue, take it away whenever you're ready.
Thank you, Sarah, for that introduction. Here are my disclosures. So first, I'd like to thank the organizers of this webinar series for the opportunity to discuss our research on skeletal muscle plasticity and aging. Recent findings show that skeletal muscle mass and strength are critical factors in maintaining an independent, active, and healthy life. The regulation of skeletal mass and strength occur over a lifetime and are dependent on a number of factors which change over time. During early life, muscle mass and strength increase rapidly and are dependent on increases in specific growth factors and hormones, as well as increased satellite cell proliferation and differentiation. In addition, increased external loading and neural activity are critical for reaching peak mass and strength. As an adult, maintaining peak mass and strength are highly dependent on the level of external loading and neural activity. With aging, mass and strength decrease in all individuals, but at different rates dependent on multiple factors, including the level of loading and activity, as well as other factors such as inflammation and metabolic stress. Factors that can accelerate the rate of sarcopenia include a sedentary lifestyle, diseases such as diabetes and obesity, and acute illness or injury, which in older individuals may be followed by incomplete recovery. This will be a major focus of my presentation. As discussed in other talks within this webinar series, loss of muscle mass with age occurs over a prolonged period of time. This histogram shows the mass of the medial gastrocnemius and tibialis anterior muscles of male Fisher-Brown Norway rats aged 6 to 30 months. One can see that these predominantly fast-twitch muscles reach a peak mass around 9 months and begin to lose mass after 18 months. In contrast, the soleus, a predominantly slow muscle, doesn't begin to lose mass until after 26 months of age. These data highlight the fact that loss of skeletal muscle mass and strength can be variable across different muscles. Another aspect of aging that we have been interested in is anabolic resistance. One definition of anabolic resistance is a decrease in the ability to activate protein synthesis in response to growth cues such as amino acids or increased loading. These data from Fry et al. Show the fractional synthesis rate of the vastus lateralis muscle following a single bout of resistance exercise. Young adults show a significant increase in protein synthesis over 24 hours post-exercise, while older adults have an attenuated response as seen here. An alternative definition of anabolic resistance is attenuated growth in response to growth cues. These data from Rao et al. Show the response of young and old females to a 12-week progressive resistance exercise program. Following 12 weeks of training, there is a significant increase in the size of the vastus lateralis muscle in young but not old females. Further examination of single fibers from vastus lateralis biopsies showed significant increases in the cross-sectional area and strength of the fast type 2A fibers in young but not old females. This table from the same publication suggests that the loss of growth response is progressive, showing that 74-year-old women have a differential response to resistance exercise training, while 85-year-old women have no response. We have examined anabolic resistance in rats using the functional overload or synergist ablation model. In this model, the plantaris muscle is functionally overloaded by removing the soleus and medial and lateral gastrocnemius. These data show the growth of the plantaris to 14 days of functional overload in male Fisher-Brown Norway rats at 6, 9, and 30 months of age. One can see that the amount of growth is significantly less in 30-month-old rats relative to 6 and 9-month-old rats. We have also looked at additional ages between 9 and 30 months and found that the attenuated growth response to functional overload occurs much earlier than 30 months of age and was evident in late middle age as early as 18 months. The functional overload model, however, is an extreme model of increased loading. So we have also examined growth in another model, the hindlimb unloading reloading model. In this model, the hind limbs are unloaded by tail suspension, resulting in muscle atrophy as shown here in the medial gastrocnemius in purple and tibialis anterior in green. A strength of this model is that one can easily remove the suspension and allow reweighting or reloading of the hind limb muscles, allowing for the analysis of regrowth as shown here. We have used this model to study the response of mature adult 9-month-old and old 28 months old male Fisher-Brown Norway rats to both disuse and reloading over periods of up to 14 days. These graphs show the response of the soleus, plantaris, and tibialis anterior muscle in adult Fisher-Brown Norway rats to unloading and reloading. Loss of mass is observed over 14 days of unloading, and upon reloading, mass increases, approaching baseline levels by 14 days. In some muscles, such as the plantaris and tibialis anterior, we also see a brief period of edema immediately following reloading. The response of these muscles in old rats is shown here by the black symbols. One can see that at baseline, these old muscles weigh slightly less than adult muscles. The amount of atrophy observed in the old rats is similar to what is seen in the adults. However, the recovery of muscle mass is significantly impaired and does not reach baseline within 14 days. We have also measured fiber cross-sectional area and find a similar lack of recovery of mean fiber cross-sectional area in all muscles. Strength of the ankle extensor muscles was also measured in vivo using the Aurora scientific system. Isometric torque was measured by securing the foot to a force plate, stabilizing the knee, and stimulating the nerve with needle electrodes. The baseline torque frequency relationship shown here in adults and old rats shows a significant decrease in maximum isometric torque in old rats at baseline. Repeated measurement of isometric torque shows a decrease in maximum torque following 14 days of unloading, seen here in red. In old rats, the decrease in torque is significantly greater following unloading, even though the amount of atrophy is similar to what is found in the adult rats. Measurement of isometric torque following reloading shows complete recovery of isometric torque in adult rats. However, in old rats, no improvement of isometric torque was observed over 14 days of reloading. A follow-up experiment extending the reloading period to 28 days showed a significant improvement in isometric torque, as seen here by the black circles. One possibility for the lack of improvement in mass and function could be a reduction in cage activity in the old rats upon reloading. We measured cage activity in adult and old rats using a beam brake system. Prior to unloading, cage activity was similar in adult and old rats. Upon reloading, cage activity was significantly reduced over the first 3 days. However, by 5 days of reloading, adult and old rats had similar cage activity. In summary, we found a similar degree of muscle loss in young and old rats. There was a greater loss of force in the old rats, however, following unloading, and an attenuated or delayed recovery of muscle size, especially of the type 2B fibers, and isometric force upon reloading. So how do these data compare to humans? A study by Sweta et al. Compared the response of young and old males to 2 weeks of cast immobilization, as shown here in these graphs. Type 1 and type 2 fiber cross-sectional area of the vastus lateralis muscle was measured, and full recovery of fiber size was observed in young adults. However, there was a lack of recovery in the old subjects after 4 weeks of reloading. A follow-up experiment by the same group examined the response of young and old subjects to 4 days of knee immobilization and 7 days of recovering with the addition of resistance exercise. Measurement of knee extensor torque showed a greater reduction in strength in the old subjects relative to the young subjects, and also an attenuated recovery post-exercise, or post-reloading, even with exercise. These data show that the hindlimb unloading reloading model in rats replicates what is seen in humans during disuse. So what is responsible for the attenuated growth and lack of functional recovery seen in old rats? We investigated a number of possibilities. One possibility is altered proteostasis. We measured protein synthesis and degradation in these rats and found an increase in mTORC activation and protein synthesis in both adult and old rats. Unexpectedly, we did not find an increase in protein degradation, but rather found a suppression of proteasome activity during both unloading and reloading in the old rats. This imbalance between synthesis and degradation could produce an increase in ER stress, which we found at baseline and in response to loading. Altogether, these data, which have been published, suggest that altered proteostasis could contribute to the attenuated recovery of muscle mass. Another possibility for the loss of strength is neuromuscular junction instability and denervation, which I will discuss for the remainder of this talk. First, I'd like to review a few aspects of the neuromuscular system. Muscle fibers are innervated by motor neurons, and each motor neuron innervates a select number of muscle fibers called the motor unit. The motor axons make contact with the muscle fibers, forming the neuromuscular junction. The neuromuscular junction is critical for activation of the muscle fiber, and a number of specific proteins, such as the acetylcholine receptor subunits, are expressed at this site. Motor units can be classified into different types based on a number of properties. In a mixed muscle, fast fatigable motor units produce the greatest tension and are composed of the greatest number of fibers, which usually express type 2B myosin heavy chain. The fast fatigue-resistant motor units are intermediate in size and strength, and the fibers usually express the type 2A myosin heavy chain. Finally, the type S units, or the slow units, are the smallest in size and strength, and the fibers usually express the type 1 myosin heavy chain. With aging, terminal axons retract from specific fibers, resulting in denervation. At early stages of aging, these fibers become reinnervated by another motor neuron, and the properties of the fibers change to reflect the properties of the new motor neuron. This process results in fiber type grouping, as seen here. These pie charts show the fiber type composition of the adult and old rats in our studies. One can see that in the mixed muscles, such as the plantaris and tibialis anterior, there was a significant change in the fiber type composition, with an increase in type 1 and 2X fibers and a decrease in the percentage of type 2A and 2B. As noted, this was particularly notable in the tibialis anterior, which has the greatest number of 2B fibers in the adult. This slide shows representative sections of the deep and superficial regions of the tibialis anterior in adult and old rats, stained with antibodies against the type 1, 2A, 2X, and 2B myosin heavy chains. In the deep region, all fiber types are expressed, and there is minimal grouping of fiber types. In contrast, the superficial region is composed of only type 2X and 2B fibers. With aging, one can see the appearance of fiber type grouping, seen here especially in the type 1 fibers shown in blue and the type 2A fibers shown in red. In the superficial region, one can also see fiber type grouping, but of the type 2X fibers, as the percentage of 2B fibers decreases. One can also see the decrease in fiber size seen especially in the 2B fibers. So is there evidence of motor unit plasticity and neuromuscular junction instability in aging rats? A recent study from Padilla et al. Showed changes in motor units with aging. In this study, they measured compound motor action potentials, single motor unit potentials, and motor unit number in the gastrocnemius of young and old Fisher 344, and showed a decrease in the compound motor action potential with age, an increase in the single motor unit potentials suggesting an increase in size of the motor units, and a decrease in the number of motor units reflecting a loss of motor neurons. These authors also examined neuromuscular junction stability. Consistent with our data, they found a decrease in strength of the ankle extensors in old Fisher 344 rats. Examination of individual contractions showed an inability to maintain force during the 1 second train, which was quantified as percent torque loss, quantified as percent torque loss. These data are suggestive of neuromuscular junction instability. Increased expression of specific genes has also been suggestive of an inactivity or denervation. We measured expression of these genes and saw an increase in the expression of HDAC4, myogenin, RUNX1, acetylcholine receptor alpha, acetylcholine receptor delta, MuSK, and NCAM in all muscles at baseline. This was especially true for the mixed muscles, the medial gastrocnemius and the tibialis anterior. We then performed an RNA-seq analysis to compare adult and old medial gastrocnemius muscles at baseline, 7 days of hindlimb unloading, and 3 and 7 days of reloading. We found differential expression of several hundred genes at baseline, and thousands of genes were found to be differentially responsive to the treatment. Further, we found hundreds of genes that were responsive to both age and treatment. We were especially interested in those genes that differed as a function of both age and treatment. In particular, we were interested in what was happening during the unloading period. Interestingly, a large number of genes were differentially expressed in response to unloading in both adult and old rats. However, only a fraction of the genes, 356 shown here, were similarly affected in adult and old rats. Many of the genes that were affected by age and treatment were those associated with inactivity and/or denervation. In the next 3 slides, I will show data from a few of the genes that were differentially expressed as a function of age and treatment. This slide shows three genes, HDAC4, RUNX1, and myogenin, all of which had significantly higher expression levels at baseline in the old rats. Upon unloading, HDAC4 increases in both adult and old rats to a similar degree. However, RUNX1 and myogenin expression was significantly higher in the old rats during unloading. Upon reloading, we see that in the adult animals, these genes decrease expression back down to baseline. However, in the old rats, expression of all three genes remained elevated, and in some cases, as can be seen with RUNX1 and myogenin, expression increased upon reloading. Here we show acetylcholine receptor alpha and NCAM. Again, both genes show elevated expression in the old rats at baseline, and upon unloading, again, both genes increase somewhat in the adult animals, however, increase significantly to a significantly greater extent in the old rats upon unloading. And again, upon reloading, these genes go back to baseline levels in the adult animals, but continue to be elevated in the old rats during the reloading phase. And again, in the case of the acetylcholine receptor alpha, increase upon reloading, increase to a greater level than seen during unloading. The next four genes have been associated with muscle atrophy. As you see, GAD45A and P21 both had elevated levels of expression in the old rats at baseline. Upon unloading, GAD45 increases to a much greater extent in the old rats than the young rats, and again remains elevated during the reloading phase. The same pattern can be seen for P21. MRF1 and MAFBOX are E3 ligases which have been associated with muscle atrophy. Here we see that MRF1 is significantly elevated at baseline, but MAFBOX is not. Upon unloading, both genes increase to a similar degree in both young and old rats. However, upon reloading, both MRF and MAFBOX decrease below baseline levels in the adult, but remain elevated in the old rats. So in summary, we found differential gene expression in young and old rats during unloading and reloading. We found an increase in the expression of genes associated with inactivity and denervation in the old muscles following unloading and reloading. And we found an increase in expressions in genes associated with muscle atrophy in old muscle during unloading, and continued elevated expression upon reloading. So does neuromuscular junction instability occur in humans following disuse? A recent study by Monti et al. Examined the response of young healthy males to 10 days of bed rest. Following 10 days of bed rest, there was a significant decrease in maximum voluntary contraction of the knee extensors. When normalization of maximum voluntary contraction to cross-sectional area revealed that the loss of strength was greater than the loss of cross-sectional area. Fibers in the vastus lateralis were also analyzed and showed an increase in the percent of fibers that stained for NCAM, a marker of denervation, and also an increase in the expression of the acetylcholine receptor alpha, a marker of inactivity and denervation. All of these data suggest that neuromuscular junction instability does occur with disuse in healthy adults and is similar to what we saw in adult rats following hindlimb unloading. Another recent study looked for evidence of neuromuscular junction instability in older individuals. They found a decrease in fiber size in the vastus lateralis, mostly in the type 2 fibers, as has been previously reported. They also examined fibers from the vastus lateralis and found an increased expression of the neonatal isoform of myosin heavy chain and NCAM in fibers of the vastus lateralis of elderly subjects. Further, they found an increase in the expression of genes associated with the neuromuscular junction and suggestive of inactivity and/or denervation. Many of these genes are the same genes that we saw elevated in our rat model. In conclusion, aging results in a delayed or attenuation of the recovery of muscle mass and force in both old animals and humans. Type 2B fibers appear to be the most impaired in the aging models. Disuse results in neuromuscular junction instability, and aging appears to enhance this instability. In aged rats, reloading appears to induce further neuromuscular junction instability and possible denervation, resulting in significant decreases in force production. These data highlight the importance of the neuromuscular system in the regulation of muscle mass and strength, and show that further study is needed to examine possible therapies to prevent neuromuscular junction dysfunction with aging and inactivity. I would like to thank you for your attention and acknowledge funding from the VA and the National Institute of Health, and also acknowledge the many people that have contributed to these data. Thank you.
Okay, Sue, first question for you. This person has asked, your experiments were done in 28-month-old FBN rats. Have you studied earlier ages?
Yes, so in the Fisher-Brown Norway rats, we have looked at an earlier age of 24 months. And at 24 months, we see complete recovery following the unloading. We've also looked at a different strain of rat, the Fisher 344, that has a shorter lifespan. And we looked at 24 months, which we thought was a comparable time age as far as their life expectancy. And what we saw there was a delay in recovery at 7 days, but by 14 days, they were fully recovered. So, and there were significant differences between the Fisher 344s and the Fisher-Brown Norway, especially in the response to some of the gene profile we saw with the neuromuscular junction. So we think there's that the earlier ages, the difference between the Fisher 344 and the Fisher-Brown Norway has to do with one, the age. The 24 months is still young enough, and that there haven't been significant changes at the neuromuscular junction or inactivity or denervation is not as big of a problem at that age as it is at the older ages.
Okay, great. Our next question here is, you showed that force improved after 28 days of recovery. Were there improvements at the neuromuscular junction?
So we did do the follow-up experiment at 28 days and saw that the force and mass was starting to recover. It wasn't completely recovered, but what we did see, we looked specifically at some of these neuromuscular junction markers, the gene expression, and what we did see is that by 28 days, many of these genes that were elevated at immediately post-recovery, the neuromuscular junction and inactivity genes had been downregulated, were back down to baseline by 28 days. So we suspect that the improvement in force was related to either stabilizing the neuromuscular junction, or if there had been denervation, there was reinnervation of those junctions. So we haven't done any further follow-up, but it's something that we're very interested in.
Great.
Thank you.
Cool future direction. Okay, the next question here is, are there changes in fiber type or number normalized to body weight and or BMI?
So the fiber type data I showed, no, we didn't normalize those data to body weight or BMI. The way the fiber type was assessed is we took multiple regions within the muscle, so up to 6, and the same regions in the young and the old in the particular muscles, and then calculated the fiber type based on the myosin heavy chain expression. And we didn't count fiber numbers, so we don't know if there was a decrease in fiber numbers at that time. Thank you.
Okay. We've got another question here. Are there any NMJ structure changes compared between young and old rats?
Yeah, that's a very interesting question. We didn't do any measurements of structural or look at the NMJ structure. It's something that we are proposing to do in follow-up experiments. I suspect there's a recent paper that came out looking at NMJ structure in young, well, that compared young and old, and there were, after unloading, and there were significant differences. So I was really excited to see that paper. So I suspect that we would see differences at the neuromuscular junction in the young and old. But it's something we're very interested in following up on.
Okay, great. Somebody here has asked, did you check the expression of any pro-inflammatory cytokines during unloading or reloading to compare between young and old animals?
Yes, so we have looked at inflammatory markers and NF-kappa B signaling and some other inflammatory markers, and we do see, these data haven't been published, but we do see elevated overall inflammatory markers in the old animals, and especially and increased activity, increased signaling within the NF-kappa B signaling. So it's possible that inflammation is contributing to the lack of recovery. One of the things we've noted, and I pointed out, is in the old animals, upon reloading, there's this lack of edema at the initial stage, and that actually may be. And there have been people that are looking at macrophage, both pro-inflammatory and anti-inflammatory macrophages in these following unloading and reloading, and so there may be that may play a role in or contribute to the lack of recovery. It's something we're very interested in again, and are looking into in follow-up experiments.
Awesome. All right. Our next question here is, you indicated that aging is associated with reduced proteasome activity. Have you measured other proteolytic pathways like lysosomal autophagy, calpains, and do they follow the same pattern?
Yes. So we did measure cathepsin L, and we've measured autophagy, and actually at baseline, autophagy was elevated in these animals, in the old animals. What was interesting is that upon reloading, it didn't go up. So, and proteasome activity was blunted in the old animals, whereas in the young animals, proteasome activity was elevated even upon reloading during the recovery period. So autophagy was high, but it didn't increase any further from baseline. And we also saw corresponding increases in cathepsin L. So one thought one of our hypotheses is that during that reloading phase, when protein synthesis is high, and it is high in the aging animals also, is that there's an imbalance between synthesis and degradation, and so you get more that synthesis is not keeping up, or sorry, synthesis is overwhelming the degradation pathways, and we're getting potentially either misfolded proteins, which are then not getting properly folded and or degraded, and so we're getting, and that's contributing to increased ER stress, which we see significant increases in ER stress upon reloading. So we do think there's a mismatch between synthesis and degradation during these reloading phases.
Awesome. Great answer. Okay. Got another question here. We've got lots of questions, so I'm going to just keep going if you're okay with that.
Fine.
This question is, would you know if there's any association between changes in the extracellular matrix and the neuromuscular junction instability?
Good question. Interesting question. I mean, we have looked at some extracellular markers, and there are significant changes in the old animals relative to the young. And so that could contribute to changes in the extracellular matrix, could contribute to neuromuscular junction stability. So it's interesting question, and should definitely look at that. Thank you.
Great. There is another question here. Do you think in an older population that has been continuously exercising for much of their lives, that they may be protected from immobilization-induced atrophy, or perhaps recover more quickly, similar to a younger individual?
Short answer is yes. I think whether they may or may not be protected against atrophy, because even the young animals that are healthy atrophy, and active, and young, and even young humans, but I think that they may have better recovery. And I mean, so the question of is experiment that we've been interested in pursuing is exercising, putting rats on an exercise program, a chronic exercise program at an older age, and then doing the unloading and reloading and seeing if they have enhanced recovery. But it's a good question, and I suspect that they do recover better.
Okay, great. Another question here. Did your gene data show any large changes in mitochondrial gene expression with reloading in aged animals?
Yes. So mitochondrial genes went up. So they decrease with unloading and increase with reloading. We haven't done I haven't looked to see if they go up comparably, but yes, they do increase upon reloading.
Okay. Another question here is, have you looked at whether changes in changes in mitochondrial components and how they can contribute to the abnormal NMJ integrity?
We haven't looked at that directly. So this is asking mitochondria at the NMJ, or can you repeat the question?
Yep, they just asked, have you looked at whether the changes in mitochondrial components could contribute to the abnormal NMJ activity?
Oh, yeah. So yeah, we haven't done a extensive analysis of mitochondria components or function in these animals. it's possible that, that there is an energetic imbalance in these older animals. And I mean, we see this very high level of protein synthesis, which we didn't expect. Our initial hypothesis was that protein synthesis was going to be down in these older rats. But one of the things you see in the older rats is even at baseline, they have elevated protein synthesis. And so upon recovery, protein synthesis increases, but the differential between baseline and recovery is not as great as you see in the young. So there still could be a problem with protein synthesis, and it may be that these animals do have some type of energetic stress.
Okay. Another question here. Your experiments were done in male rats. Do older females have the same attenuated growth response?
Yeah, so we've been interested in that question initially. So our initial experiments were in the Fisher-Brown Norway strain that we received from the NIA's rodent colony. And unfortunately, we couldn't get the older females in that strain. So we did do some experiments in the Fisher 344s in both males and females. And initially, we did the females, but because the Fisher 344 has a shorter lifespan, we couldn't go out that long. So we did 24 months, which we thought was equivalent to 28 months in the Fisher Brown Norways. And we saw that the females had full recovery. And that's when then we thought, oh, maybe there was a male-female difference. And that's when we did the males in the Fisher 344s and saw that they also had full recovery. So we think it's a strain difference and not necessarily a gender difference. So we would like to do, now that the NIA rodent colony has the older Fisher Brown Norways females, we're proposing to do some of the experiments in the females at an older age.
Okay, great future direction there. All right, this next question is, are there any changes in blood flow to hind limb muscles in the unloading model?
So we didn't measure blood flow. I'm sure this data is out there in this model. I suspect that there is some decrease in blood flow, but these the limbs are still active in this model, so the animals still kick their limbs, and the muscles are still contracting. So it's not complete inactivity. So blood flow may be reduced. I suspect it's reduced relative to if these animals were full weight bearing and walking, but they do still actively contract the muscles when they're in that in the tail suspension. So.
Okay, great. Our next question here is, aging is associated with chronic inflammation. Do you think that there is a change in inflammation markers with unloading and reloading in mice?
So yes, I mean, we've done it in the rats. So we do see elevated markers in the older animals with inflammation.
Okay. Another question here is, do you have any data on agrin expression?
We do. I know I looked at it. It's in our RNA-seq data, and I can't remember if it was up or down. I know agrin's associated with the neuromuscular junction, and we do see many, like musk and other, was elevated. So I can't remember what agrin did. Sorry.
That's okay. This person has asked, in your opinion, what is the most promising future direction in this type of research?
Oh, wow. I mean, I think there are many. I think trying to understand what is inhibiting the recovery. I don't think it has gotten enough attention. I think there's definitely, I think the what's happening at the neuromuscular junction and activation of muscle is definitely an area that's getting more attention, and I think there's considerable data now, both in animal models and humans, that this is a problem. So I think this is a very promising area. Um, I think there's a lot to look at with respect to proteostasis and the balance between synthesis and degradation. I think one of the things we don't know is, even though synthesis is up in these animals, is what are the specific proteins that are being synthesized. So it may not be the correct ones. And I do think, even though proteins may be synthesized, they may not be folded properly, and the building back of the myofilaments may be impaired. So the proteins may be there, but then putting them, back into the reconstructing the sarcomeres and the myofilaments and the myofibers may be impaired. So I think looking at that more closely is definitely an area that I don't think has gotten a lot of attention.
Okay, fantastic. And I think, just based on time, we're going to make this our last question.
Okay.
So this question, well, it starts with, thank you for an excellent talk. And the next part of the question is, with regard to the gene expression aspect of your work, have you or do you plan to investigate the role of epigenetic modifications on gene expression?
We are collaborating with a group in Norway and looking at epigenetic changes with aging and recovery. So it is something we are interested in, and definitely collaborating with, or have a collaboration with Adam Sharpless in Norway. So it's an interesting area to pursue.
Yeah, definitely. Well, I just wanted to thank you again for all of your fantastic insights today. It really was a pleasure to have you with us.
Thank you very much, Sarah, and for allowing me the opportunity to speak.